Our next speaker will be Summer Page, who will be discussing understanding the placebo analgesia on a neural level. Thank you, Summer.
Hi. Hi, thank you for this opportunity. Can you see my screen all right?
Yes, that's fine.
Awesome. Hi, I'm Summer, and I'm studying placebo analgesia and looking for a neural basis for this in rats modeling chronic neuropathic pain. So chronic pain is a significant health burden in Australia, and current treatments lose efficacy over time and cause significant side effects. Now, placebos have been shown to treat chronic pain as effectively as active drug treatments, but individuals vary in their responsivity to a placebo treatment. Placebo reactivity may in part be due to activity within the periaqueductal gray, or the PAG, particularly in the ventrolateral column, which hosts the descending analgesic pathway. So this lab previously elicited placebo analgesic reactions in male and female rats following chronic constriction injury of the sciatic nerve. So rats underwent 4 days of morphine conditioning with cold plate testing, and the cold plate was housed inside a conditioning chamber, seen here, which had several sensory cues, such as the fractal wallpaper we can see, as well as a vanilla scent, which the rats learned to associate with the pain relief that they felt from the morphine treatment. So on a final test day, morphine was replaced with saline, and the researchers observed that rats could be split into 2 groups based on their behavioral responses. So in one group, rats displayed significant placebo analgesia. So they displayed fewer hind paw withdrawals on the cold plate, as we can see in the blue boxes here. Whereas in the other group, those sensory cues in the conditioning chamber didn't elicit any placebo analgesia. So this left the question of what differentiates the placebo reactors from the non-reactors. And in my study, I examined the neural activity of these rats within the periaqueductal gray. And I expected that in the ventrolateral column, the placebo reactors would exhibit higher activity, indicating that that descending analgesic pathway was active in producing placebo analgesia. So to do this, I immunohistochemically labeled the midbrains of these rats from this conditioning paradigm for CFOS. And I quantified CFOS positive cells and compared between placebo reactors and non-reactors, as well as by sex, side, rostrocaudal level of the PAG, and staining intensity of the cells. Now, rather than seeing that placebo reactors had higher activity, I actually observed no differences in the females, as we can see in B here, and the males showed higher activity amongst the non-reactors, particularly in the caudal region, and amongst the lightly stained cells, as we can see in these figures here. So these results indicate that in placebo non-reactors, normal neuronal activity in the PAG in the ventrolateral column is disinhibited when the drug is removed. And this may be what blocks placebo analgesia from being elicited by the conditioning procedure. This heightened activity in the ventrolateral column here might also indicate a high prediction error in those placebo non-reactors. And the fact that this occurred only in males suggests that there may be sex differences in the descending influences on PAG activity, which might contribute to placebo analgesia. Thank you very much.
Okay, great. Thanks a lot, Summer. So next up, we have Amira Skeggs to talk to us about cultural and linguistic influences on frontotemporal dementia. Thanks, Amira.
Good afternoon, everyone. Let me just share my screen. Can you see that?
Yep, all good.
Good afternoon, everyone. My name is Amira, and I am an honours student at the Frontier Frontotemporal Dementia Clinic at the University of Sydney. How does culture influence dementia? That was the question at the heart of the present study, which aimed to examine whether the dementia profiles of culturally diverse patients differed from those typically reported in Western populations. In this study, we aim to examine differences in the clinical features, verbal and non-verbal cognitive test scores, and social cognition of culturally diverse and non-diverse patients with frontotemporal dementia. Frontotemporal dementia is an early onset dementia characterized by atrophy to the frontal and temporal brain regions. Here, we compared 3 behavioral variant frontotemporal dementia groups. These were Australian-born, foreign-born and spoke English as a first language, and foreign-born and spoke a non-English first language. A control group of healthy Australian adults was also included as a reference point for normal cognitive function. We found evidence that culturally diverse patients presented with different clinical features. Specifically, foreign non-English patients showed greater disinhibition and hyperorality at clinical presentation. In contrast, Australian patients presented with greater apathy and loss of empathy. Foreign non-English patients were also diagnosed approximately 2 years later than Australian patients, demonstrating a delay in diagnosis. On cognitive tests, Australian patients performed poorly on verbal and non-verbal cognitive tasks. In contrast, both foreign groups only performed poorly on verbal tasks. A similar pattern of results emerged on tests of social cognition, where foreign patients performed poorly on the verbal affect selection task, but not on the non-verbal discrimination task. As we found evidence of testing biases even for foreign patients that were English speaking, we ran additional cultural analysis on cultural factors that were not related to patients' first language, such as years lived in Australia and years of English education. Overall, we found that less acculturation to Australian culture was associated with poorer performance, both on verbal and non-verbal cognitive tests. This suggested to us the presence of cultural biases in the present tests that occurred irrespectively of their verbal demand. To conclude, culturally diverse patients showed evidence of a milder clinical profile than Australian patients. This was likely explained by the fact that all these patients were bilingual and also more educated, two factors which are known to have a protective effect against early cognitive decline. As predicted, we also found evidence of verbal testing bias for culturally diverse patients. Interestingly, however, we also detected cultural biases, which meant that even culturally diverse English-speaking patients suffered a cultural disadvantage during clinical assessment. Overall, these findings implicate both cultural and linguistic biases in current clinical tests, challenging the validity of these assessments for the future assessment of culturally diverse patients. Thank you very much.
Thanks, Amira. Our next speaker will be Carissa Lee, who will be discussing white matter changes in frontotemporal dementia. Thank you.
Alrighty, can you all see my screen?
Great, thanks.
Amazing. Alrighty, hi everyone. I'm an honours student with the Frontier Clinic. So frontotemporal dementia, or FTD, is an extremely complex disease. Pathological and clinical heterogeneity means we can only accurately determine the pathology underlying FTD post-mortem. An accurate biomarker of underlying pathology in vivo would improve early accurate diagnosis, monitoring of disease progression, and the development of clinical trials. So our study aimed to examine the white matter changes associated with the two main pathology types of frontotemporal lobar degeneration, which are tau and TDP43. We investigated this in a clinically heterogeneous cohort of FTD cases with both post-mortem pathological confirmation and diffusion-weighted MRI data. A novel pixel-based analysis, which has improved accuracy and biological interpretability, was used to quantify white matter changes. Our analyses examine white matter changes within and between pathology subgroups, controlling for disease severity and clinical presentation. So overall, we observed distinct neuroanatomical patterns of white matter changes between tau and TDP43 patients. White matter degeneration affected primarily frontotemporal regions in tau and motor-related regions in TDP43 patients. These pathology-specific changes parallel different clinical atrophy patterns, reflecting the phenotypic characteristics of the pathology groups. Distinct pixel-based metrics of white matter changes also differed in the region and magnitude of effects. So our study was the first to directly examine the relationship between white matter changes and underlying neuropathology in a cohort constituting the entire clinical spectrum of FTD. Our findings suggest white matter imaging is a promising biomarker of underlying pathology in vivo. This has implications for the outstanding challenges faced in FTD. As white matter atrophy is proposed to be one of the earliest pathological changes observable in FTD, our findings indicate white matter imaging may be used to improve early accurate diagnosis, even before symptom onset. Our findings also suggest that pixel-based white matter imaging can inform the trajectory and mechanisms of the brain changes in FTD, which are presently not well understood. Most critically, no effective pharmacological treatment is presently available to patients with FTD. Our findings can inform the development of clinical trials of disease-modifying treatment, as white imaging could serve as an early surrogate outcome measure and provide the earliest evidence of treatment efficacy. Thank you.
Right, thank you very much, Carissa. So our penultimate speaker is Emily Joyce, and she's going to talk to us about neuropathic pain. So over to you, Emily.
Can everyone see that?
Yep.
Great. Hi everyone, my name is Emily, and today I'll be speaking about photobiomodulation, or PBM, which is a treatment that involves shining a red or infrared laser over injured tissue, and it's been found to offer profound pain relief in neuropathic pain settings, an area where other pain treatments lack efficacy and suitability for long-term use. What we're really interested in within our study is looking at the impact of PBM on the immune system, and specifically macrophages and CD4 T cells. These cells can be divided into smaller subsets based on pro or anti-inflammatory cytokine signatures. So our pro-inflammatory M1 and Th1 cells produce cytokines like TNF alpha and IL-1 beta, which drive neuronal signaling, neuronal firing and pain transmission. On the other hand, our anti-inflammatory subsets like M2 and Th2 cells produce anti-inflammatory cytokines like IL-10, which are able to dampen down that pro-inflammatory immune response. So we wanted to look at whether the pain relieving effects of PBM can be attributed to its driving of the polarization of cells towards this anti-inflammatory phenotype. So to model neuropathic pain, we used a chronic constriction injury, and this is able to effectively induce mechanical allodynia, which is pain occurring from a normally innocuous stimuli, and thermal hyperalgesia, which is where a painful stimuli is perceived as excessively painful. To give the rats their daily PBM treatment, the laser was shone directly over the injury site for the 14 days, and the animals were also being tested for their mechanical and thermal pain sensitivities during these 14 days. So looking at our results here, our PBM treated group is shown in red, our untreated group is shown in yellow, and our sham group is shown in blue here. So what we can see is that while PBM treatment was mildly effective at alleviating mechanical allodynia, it was really effective at alleviating thermal hyperalgesia, which you can see by this return to baseline levels here. And a reason behind this difference could be due to PBM's upregulation of endogenous opioids, which has previously been found. And the reason that this would attenuate hyperalgesia more than allodynia is due to the different pain pathways behind these symptoms. So hyperalgesia is carried by nociceptive C and A delta fibers, which express opioid receptors, whereas allodynic inflammation is carried initially by AB mechanosensitive fibers, which don't express the receptors to the same degree and therefore wouldn't be inhibited by beta endorphin to the same degree, meaning allodynia was attenuated to a lesser degree. Interestingly, when we analysed the peripheral blood cells from these animals using flow cytometry, we didn't see a large modulation of monocytes or CD4 T cells in terms of their polarization towards pro or anti-inflammatory subsets. And this was surprising, given that the anti-inflammatory polarization of PBM is known on tissue-invading cells. So it suggests that tissue-invading macrophages and CD4 T cells may be more implicated in PBMT-mediated pain relief. Thanks very much.
Thanks very much, Emily. And our final speaker for the session is Timothy Wong, who will be talking about the relationship between executive function and social cognition in autism. Thank you.
Hi everyone, can you see that properly?
That's great, thanks.
Okay. So those on the autism spectrum experience varying degrees of social impairment. Often this means having difficulties communicating and engaging in social interaction. So the goal of my project is to determine which mental processes predict social impairment in autism and to determine how those processes are related. So just briefly, most of us can recognize emotions and reason about other people's thoughts and feelings quite intuitively, and this is known as social cognition. Again, most of us can inhibit our impulses and direct our attention flexibly to achieve goals, and this is referred to as executive function. And we know that autistic people, especially autistic children, score poorly on measures of executive function and social cognition relative to neurotypicals, which means that these mental abilities could explain social impairment in autism. And so what I hypothesized was that executive function scores would predict higher order social cognition, so being able to reason about other people's thoughts and feelings, but not lower order social cognition, so primarily emotional recognition in autistic adults. So how did I do this? I analyzed data. So I analyzed data from 140 treatment-seeking autistic adults, assessing them on higher order and lower order social cognition. Crucially, the first aim of my study was to see whether these executive function measures over here would predict these social cognition measures over here. And so what did I find? I found that having a high IQ and being male are associated with better performance on the eyes test. So this is a test that looks at how well one can recognize emotions from the eyes. However, executive function didn't translate to better performance on the eyes test. I also found that having a high IQ is associated with better faux pas recognition. So the faux pas test is a fun little test that assesses how well you can understand socially awkward situations. However, and again, we found that better executive function didn't translate to better performance on the faux pas test. So what are the takeaways? Overall, executive function and social cognition seem to be quite separate mental processes in autistic adults. And this is quite interesting because previous literature shows that they are related in children. And so this change between childhood and adulthood could be because the brain undergoes changes between childhood and adulthood, or because the tests for these mental processes are different between children and adults. So what this implies is that autistic adults, should they seek functional training, might need training that targets executive function and social cognition difficulties separately. And if I was to design the study from scratch, I would use more measures for social cognition and executive function, look more closely at group differences such as sex differences and presence of comorbidities, and include a neurotypical control group. Thank you.
Okay, so that brings us to the end of the poster session, which again showcases the diversity of cutting edge research that's being done here in the BMC, largely driven by most of the postgraduates and early career researchers. And you can view their submissions online in a poster booklet that's going to be available for download from the BMC website. So thanks again to all our presenters. I'm going to now hand it back to Leanne to announce the winners of the poster session and also finish off with some closing remarks for today. Thanks, Leanne.
Thanks, Anand. Well, what an amazingly huge cross-section of papers that we've had here today. I think Mark Scott was spot on this morning when he said the brain is the most complex thing in the world, and I think we've seen that in the presentations that we've had so far to date with our symposium, and I'm just blown away by how brilliant so many of our young researchers are, and just how clever they are. So I really would like to congratulate all our speakers today. We do have a number of people who were entered into a competition with the posters, and we had 40 entries, and they were all outstanding, and there was really very little in it between the different categories, and so I mean, I wish we could give you all a prize, really, because you were just also fantastic. I also want to thank the EMCR researchers from BMC who helped us with judging those entries. So I'm very happy to announce the Symposium poster winners for this year. So the honours winner is Summer Page, who is from the School of Medical Sciences, and she receives the award for her paper, The Sugar Pill Enigma, Understanding Placebo Analgesia on a Neural Level. So congratulations, Summer, on an excellent poster. Moving on to our HDR category winner, that is Susan Lin from the Sydney Pharmacy School, and she presented a poster, A Simple Model of Desensitisation for Analysing Epilepsy-Associated GABA Receptor Variants. And our final winner, I think this one is the ECR category, and the winner is Dr. Marshall Dalton from the School of Biomedical Engineering for his paper Anatomical Connectivity Between Cortical Brain Areas and the Anterior Posterior Axis of the Human Hippocampus, New Insights Using Quantitative Fibre Tracking. So congratulations to all our winners, but really congratulations to all of you. The winners will receive a gift card to the value of $200. And as Anand mentioned, you will be able to view all the poster submissions that are going to be made available in a booklet on the BMC 2021 Symposium page. So congratulations. So that is our session done for today. We do have another session tomorrow, which will actually start tomorrow afternoon. So we're commencing at 1:30 tomorrow. And very excitingly, I don't think I've ever done this before, we'll be welcoming the Honourable Julia Gillard, our former Prime Minister, to address our BMC Symposium as our keynote speaker. And then we will also have a split keynote session that's going to be hosted by Professor Ian Hickey. And that's really a session that's going to focus on translation. So we're going to welcome Professor Yun Hee Jeon from nursing and Professor Jacqueline Troy. We're also going to have another full EMCR speaker session and a panel discussion, which really looks very, very interesting. So I look forward to welcoming you all again here tomorrow. I think we've had a very nice introduction to what is going to be a brilliant remainder of our symposium. So thank you very much, and enjoy your afternoon, and we'll see you tomorrow.